Hi, everyone. In conjunction with the European Research Council, EURAXESS North America is interviewing Dr. Carey, an American neuroscientist living in Europe, for the latest edition of the ERC Magazine. Dr. Carey, welcome, and could you please tell us a little bit about yourself and your project?
Hi, I'm Carey, and I'm a group leader in the neuroscience program at the Champalimaud Center for the Unknown in Lisbon, Portugal. I'm originally from Philadelphia, but I've moved around a lot. So after university at Wesleyan University in Connecticut, I moved to California, where I did my PhD at the University of California, San Francisco. And then I was a postdoc at Harvard Medical School before I moved to Portugal. So I've been in Lisbon for 10 years now, almost exactly 10 years, and it's actually the longest I've ever lived anywhere since I graduated from high school. The goal of my lab is to understand how activity is orchestrated within neural circuits to control learned and coordinated movement. We focus on the cerebellum, or the little brain in the back of your head, that has very well described cell types and circuitry. And we focus on motor control because it's a brain function that we don't have to guess about. We don't have to guess what the animal is thinking, but instead we can really directly measure the brain's ultimate output, which is movement. I've been really fortunate and I've had 2 ERC grants. For my starting grant, we established a way to measure mouse locomotor coordination and really understand specific ways that the cerebellum contributed to coordinated walking, because lots of areas of the brain work together to control locomotion. And when movement is well coordinated, different parts of the body move together at discrete times. And we found that the cerebellum was really particularly specifically important for controlling the relative movement of different parts of the body. So making sure that if they're supposed to move together at the right time, that they do that, and also making sure that moving one part of the body doesn't have unintended consequences on the control of other parts of the body that the animal might be trying to keep still. And in order to do this, to sort of keep one part of the body still while you're moving another part, it turns out that it's really useful to have an idea of how moving one part of the body will affect the rest of the body in the future. And in order to make that prediction before the movement actually happens, the brain, and really specifically the cerebellum, continuously learns to predict the consequences of moving different parts of our body, and specifically the consequences that that will have on how we need to control other parts of the body. And so then the cerebellum can generate corrective commands or calibration signals that it then sends out to the muscles to allow muscle contractions that can counteract these predicted consequences of movement. Now for my consolidator grant, which just started a few months ago, we're actually studying this learning process, how the cerebellum learns to make these predictions by perturbing ourselves, the environment that the mouse is walking in. And then we record and analyze what happens in the brain as the animal starts to re-coordinate its movement for this new walking environment. And we do this specifically by putting the mice on a little treadmill where we can actually control the belt underneath the mouse, and we can have it move faster under one side of the body than on the other side. And this makes the mouse sort of limp a bit in an uncoordinated way. And we can investigate by recording activity or perturbing activity in specific classes of neurons in specific places in the brain. We can then investigate the signals that are both going into the cerebellum to signal that something is wrong or uncoordinated about the walking, and also to study the specific neural pathways out of the cerebellum that are able to send those calibration signals to the spinal cord and ultimately out to the limbs. Although this is all very basic, fundamental research in mice, and it's not directly linked to a particular brain disorder, the brain circuitry controlling locomotion is really quite highly conserved. And cerebellar damage leads to a very characteristic loss of motor coordination across all vertebrate species. The more closely we look at mouse locomotion, the more parallels we're finding between cerebellar control of locomotor coordination in mice and in humans. So we're hopeful that these studies may not only improve our understanding of how the brain coordinates movement, but also someday help develop therapies or devices that could help restore coordinated movements to patients with various movement disorders.
Could you give some insights on how you went about putting together your project proposal? Did you find anything to be challenging?
So an ERC proposal has multiple parts. There's the institutional part, which thank goodness our grants office took care of for me. And then there are the two parts of the scientific proposal. There's a short version for the first round of evaluation and a longer version that is sent out to reviewers if you make the first cut. And I know a lot of people struggle with how to prepare two versions of a scientific proposal simultaneously. For me, it's just worked out very well to focus on the short version of the proposal. And so I sit down to write it, and I write it, and inevitably it's way too long. So I finalize it, but I save all of that extra text. It's usually methodological details and things that I have to cut out in order to stick to the length limit for the first part of the proposal. And so then after that's finalized, I take all that extra material and I incorporate it back in, and then that usually makes up the longer half of the proposal. And this last part, fortunately, doesn't actually take very long. So when I find I haven't had much time left until the deadline, I can still manage to get it done. And I don't stress about the longer version nearly as much because it isn't even opened until you pass through to the interview stage. So you'll always have a chance to defend anything that's written in the longer version of the proposal. Whereas the short version has to stand on its own together with your CV in order to get you through to the interview.
What do you think are the benefits of applying for these funding opportunities in addition to the availability of financial resources?
In terms of the benefits of applying for ERC grants, I think obviously the money is the most important part, but I actually do think that ERC grants specifically can be quite exciting to write. It's really a chance to think big about a 5-year plan of your dream experiments, but it's really not just a daydream. So I really enjoy the process of taming that initial creative vision and shaping it into concrete aims and experiments, and then figuring out how those are going to fit together to make up the full body of the proposal. I find that process really satisfying, and it always helps me clarify my thinking and gives me new ideas. So then, of course, getting the funding and being actually able to execute the project is the very best part. But even if a proposal that you're really excited about fails and doesn't get funded in that first attempt, at the very least, it's great starting material for future grant applications.